Hello and welcome to the Test Tube podcast. I am your host Dan.
Heyo, my name is Eric. We are representing Exeter in iGEM, the biggest synthetic biology competition in the world.
We are here to interview fantastic researchers and demystify the world of synthetic biology. Let's get right into it.
Woohoo! Welcome to Test Tube podcast.
Today we have the lovely iGEM Sheffield on with us. So if you both would like to introduce yourselves.
I just waved at the camera as if the podcast can see us, but hello, I'm Isaac.
And hello, I'm Brooks.
You also just waved.
Yeah, it's
been a day.
Brilliant. So if you just want to get straight into it and give us an overview of your project.
So we're doing a project which involves something called in vivo directed evolution, and we're optimizing a bunch of genetic parts using this funky new organism called Vibrio natrigens, and we're going to make a system where you can basically evolve any protein you want automatically in a bioreactor within these cells.
Because you're also talking about the evolutionary aspects of your project. Could you give us a quick overview about that?
When we were designing the project, stumbled across this field of directed evolution, which is something that people have used in the lab for ages to, sequentially improve proteins, much like proteins and genes themselves have improved over natural evolution, of course. As you know, it's the same thing. You have variations caused by usually mutation in the population. So different bits of the code are slightly different that make the proteins act slightly differently. And then a certain set of those differences may be particularly advantageous, or they'll be selected for. And I guess that's the crux really, is that evolution has the two parts, the mutation and the selection.
And what sets our project apart from many of the existing directed evolution efforts is that kind of recently it's become more possible to do these things in vivo, so in living cells. And then on top of that, the extra little adjectives you can toss in for continuous in vivo directed evolution. The continuous part there means that the whole system can operate, generate the mutations, and select for them independent of a scientist needing to be involved.
Perfect. It would probably be really useful actually for our audience if you could go through and just quickly give an overview of what mutation is, both on just like a sort of whole organism level, as well as more on a molecular.
I always think it's an interesting word because people associate the word mutation with something quite sinister. But mutation is essential to the development of life. And all it means is that every machine in our body and in any organism, most of them are made of proteins. And those proteins are encoded in DNA. I think you've talked about DNA a bunch on your podcast before, but DNA, obviously a code consisting of 4 letters. And every 3 of those letters codes for a little building block of these machines that perform a particular function. And if you change one of those letters, you may change the building block in a certain way. And because all of those building blocks of those machines have kind of unique chemical properties, you can start to change the function of the protein slightly or alter it. Maybe that makes it better, which means the protein is improved, or perhaps it makes it not good at its job at all. In that case, then the protein wouldn't be able to work and likely would be selected against. So all the mutation is it's just a change in the underlying code of the machines that run life, I guess.
Brilliant. So this change in the base code is what we would describe as variation. And most people have heard of the concept of natural selection. Variation is basically the crux of what makes natural selection work. You've got loads and loads of different variants, and the variant that is the fittest or the most successful at replicating will increase in a population, and that's what we'd call natural selection.
So for your project, are you directing your mutation for a specific protein?
So that a big part of our project originally was we wanted to make the whole thing modular. So our aim was to basically make these plasmids modular in a way that people can get hold of them, and then clone their own gene in and clone their own kind of selection mechanism in. And then, we've optimized the parts to work with any gene. But in terms of a specific gene, we're going to hopefully do a few proof of concept things if we get time with a bacterial lactose synthase gene. Originally, that was just born out of the fact that there's no vegan way to make lactose currently. It all uses animal products. And I think one of the members on our team is a huge fan of milk stout beer. typical us Northerners, we wanted to try and find a way to get a single gene that synthesizes lactose into like the yeast that would make beer. So to do that, we found a single gene that makes lactose from a bacteria. Do you remember the name of the species? Neisseria, is it?
Yes, I think it's a meningitis bacteria.
It is a meningitis bacteria, but we'll see how that goes. So yeah, we basically found a gene for that, and we're going to try and optimize that, because that gene currently makes lactose, but it also makes a few other sugars as side products. So as a proof of concept for our directed evolution system, we're going to try and optimize it to just make lactose.
Yeah.
So, I mean, last week we spoke to Vienna, who are making bricks. You guys are making beer. Our projects, oh God, we chose the wrong thing here, Eric. What would be really cool is if you guys could explain the mechanism by which you're forcing these mutations.
Two systems that we're going to try and use. The first one is called Muta-T7. It uses something called a T7 polymerase, an RNA polymerase, and that's the enzyme that reads DNA and makes the mRNA code, which then gets translated to the proteins. The reason that you specifically use this T7 one, which is originally co-opted from a bacteriophage, a virus, the promoters that you use for your gene that you're going to mutate will be specific to that T7 polymerase. And so then when you throw your gene in with your bacteria and the polymerase that they express, the mutating T7 polymerase, which we'll explain how that works in a second, will only target your gene of interest. So that's the first thing. You've got a gene with promoters that only respond to T7 polymerase. And then the second part is that the T7 polymerase is fused to another enzyme, a base deaminase, which basically rips a bit of chemistry, that's vague, isn't it? It rips a few atoms off of one of the bases, the building blocks of the DNA, and changes them, i.e. mutating them, so that as this polymerase is reading the DNA code, it will make point mutations along the way. Pairing that with the fact that the polymerase you're using is specific, you've got a specific hypermutation system for your gene of interest. So that's one way that we're going to generate some mutations. That's better characterized than the bizarre one that Brooks is going to tell you about.
We've kind of found this cool technology called HiScribe. It works by repurposing a bacterial retron, which is another fancy sounding word, but basically all that is it's a bit of DNA that gets transcribed, turned into RNA, but it folds into this kind of weird structure. So it folds back on itself and forms some unique recognition site. And then some reverse transcriptase can come along and turn parts of that RNA back into a single-stranded piece of DNA. So we're thinking about ways to increase the mutation rate there. In a paper, we've seen people also using these same base editors, but connecting those perhaps to this reverse transcriptase, so doing that editing there. Essentially, the advantage of this slightly more complex system where you're turning DNA into RNA into single-stranded DNA, then putting it back in the genome, is you can target any region where you can design the homology for. So you don't need to have promoters present. You can target any arbitrary sequence. So you can mutate a wider range of genomic loci.
I can think of one question that's probably the most sort of important for this is these sequences that are being mutated, they will constantly mutate. It's continuous mutation, as you said. How can you prevent this mutation happening? Or how can you, if you're optimizing a sequence, how can you ensure it's being optimized? We need to use natural selection. So these mutations will need to provide a survival or a fitness benefit. So a lot of these sequences hopefully will, but how can you ensure that you are optimizing them?
This was a long discussion that we had at the beginning, because this is the crux is the thing to make directed evolution in vivo, i.e. in an organism working, is you need to somehow program the bacteria to grow faster when your gene or whatever you're mutating is getting better in your eyes. There's a couple of ways to do this. You could positively select, i.e. the ones that have a better gene will gain an ability. One example, if I use the example of optimizing lactose synthase, our lactose synthase gene is being mutated, and as that's happening, it's producing either not a lot of lactose or some lactose or a lot of lactose as it gets better, or So what we're going to do is we have our mutation kind of bit of our plasmid where our gene of interest is being constantly hypermutated. And on the other end of the plasmid, we're still working out the exact on which plasmids they're going, but on, somewhere else on another plasmid or on the same plasmid, you're going to have a gene that is able to metabolize a different sugar source. We've chosen sorbitol, a secondary sugar source, so that when these bacteria are growing, if they're expressing, and I should say the sorbitol metabolism gene will be under the influence of a promoter that responds to lactose itself, the lac promoter. And all that basically means is that the gene for metabolizing our extra yummy sorbitol is only switched on when there's more lactose present. And that will only happen when our mutating gene gets better. So as the gene gets better, it makes more lactose, which means it's able to switch on the secondary metabolism source for sorbitol, which means those bacteria will not only be able to metabolize the glucose we give them, but also the sorbitol we give them, and they will outcompete the ones that can't metabolize the second sugar. Therefore, they'll grow faster, and the ones that gain better mutations for making lactose will outcompete those. And after a week, hopefully our lactose synthase gene has got pretty hench.
Yeah, and then the second approach to this, because the potential limitation of using like this very traditional biosensor regulates promoter module is that sometimes it can be a little bit tricky to connect those two. So I guess in this case, the lac promoter is very well characterized. An alternative to that then is to use some growth slowing genes, which slows the whole rate of growth of the cell. It slows everything down a bit to a crawl. So using this way to tune fitness, the idea here is that instead of turning on this gene, speeding up growth like it does with, the sorbitol breaking down gene, the sorbitol dehydrogenase, what we would have here is a constitutive promoter that expresses this growth slowing gene. So their growth is stuck at a bit of a slow rate for a while. But in front of this gene, we found a riboregulator, so a piece of RNA. And this piece of RNA normally forms some weird stem loops, so some like secondary structure folds back on itself in a way that will stop transcription of the gene downstream when another piece of RNA binds to it. And that triggers, so say more lactose triggers the transcription of more of this regulatory RNA. That regulatory RNA binds to the RNA region upstream of this growth slowing gene, and then that changes its structure so that it is no longer transcribed or no longer translated. Instead of turning on the gene when you're detecting lactose, you're turning off a growth slowing gene when you detect lactose.
But how are you finding organizing your team and putting them into like a wiki, graphic design, social media, lab? How's that working for you?
By far harder than any of the bioscience. But yeah, I think we're getting there. We've had a team that's been a bit hard to pin down because the pace of things has been quite quick over the summer in terms of developments and how little bits of direction change. Sometimes when people miss a day or a few days, we have to catch them back up. But I think we've been getting better at it.
We even got a mathematician.
Oh yeah, that's actually been very helpful.
To him, it makes sense. To me, it looks like magic, but
we have similar. One of our teammates, he's a natural scientist, so he works with math stuff.
Yeah.
Mm-hmm.
And he's made this amazing model that we now use. And we're looking at the model and he's going, yes, this is very simple. Here's how it goes. This, and this. And we go, yes. There's a numbers. There's a letter there.
He did like pages of modeling in just a few days.
Yeah, it was quite scary. Nice. It was kind of like watching a man descend into madness and then pull out this brilliant model.
Yeah, that does seem the theme, doesn't it, with iGEM, just watching us all descend into madness.
And then pull out with something brilliant.
Yeah,
hopefully.
Yeah. We had a request from our Instagram asking if we could talk about the day in the life of the average research scientist. And I think both of you would be brilliant to go into telling us what your average day as a research scientist is.
Well, it always starts off with some kind of existential crisis. I think maybe 8:30 till like 9:15.
Yeah, I usually book in at least 45 minutes for the crisis.
And how much coffee do you have in the morning?
I've had to stop drinking coffee now. That's a whole other podcast.
We have a daily question with ours, which is everyone walks in and Eric comes and we go, Eric, you got your coffee?
Yeah.
No, not today.
Yeah. Sometimes I just walk off and they're like, oh, he's going to get coffee. They just know.
So we have Eric's essential crisis in our coffee. What happens?
Yeah. We had this question when we did outreach as well. It seems like people don't really know what scientists actually do all day.
What do you mean? We stand about in white coats and tap a clipboard?
Yeah,
exactly.
Yeah, it's great. I mean, the day in the life for us at the moment has been, it's the strangest combination of being super organized and planned, but also planning for all of your plans to completely go wrong. It's balancing those. You have to have a certain, or you have to at least develop a certain sense of stability.
Yeah.
I think.
And there's a bit of a tricky balance as well, because the most common, I feel like one of the things that people forget about is a vital part of research, particularly when they're, imagining lab coats and tapping clipboards, is the reading that goes into it and actually catching up with the work that other people have done, because research is obviously a very collaborative endeavor. So, often at the start of a project, when you're researching something, you'll spend a lot of time reading about something and making plans before you even start in the lab. But something that I have kind of come to learn is a really critical skill is knowing how to balance that reading, that research, that exploration of the literature with actually starting and running experiments of your own. It's hard to know when you should just keep reading about something and when it's worth setting up an experiment yourself and just getting into the lab. And of course, the flip side of that is once you do find something cool in the lab,
Write it down.
Yeah, write it down and communicate it clearly with, everyone else. So you're giving back to that community of knowledge. And keep in mind that community of knowledge is something that you yourself will almost certainly need in a few weeks when you're trying to think back to, oh, well, what did I do this day? Or this worked really well. How much of this did I add to this flask? So
it was, we would completely agree. Earlier today, we were sat up in the breakout room of our lab, and we have Ollie and Prina who are working on some great stuff, but they're working through, and Prina said something very calmly. Ollie goes, write it down, quick, this is great, this is gold.
Yeah, anything you say, you got to write it down. Even if it's just like a quick idea and you try it, you've got to write it down, because who knows, that little idea might grow way more, who knows?
Yeah,
it's
exactly.
Definitely agree with the day of the life of a research scientist is mostly planning things that are going to go wrong.
Yeah.
So you have to make more plans, and you make those plans by reading.
Yeah, exactly.
Yep.
That sounds about right to me. That should be on the job description.
Yeah. But in case, that job description does sound too grim, I also think it's worth pointing out that at least the reason I think research is so cool is, at least at this level, once you're in university, it's almost startling how quickly you bump up against the boundaries of all human knowledge, because at this level, almost somewhat easy to try something that somebody has never tried before, which is quite an exciting feeling to know that, oh, we're entering the unknown now. And what we're essentially doing is trying to draw a nice map of the landscape that we can pass on to everyone who wants to walk these trails behind us. So there we are. There's a nice
such
poetic
a beautifully
thing.
put metaphor. That's really good. But no, it's been absolutely lovely speaking to you guys.
You
and
too,
to any listeners, go and check out iGEM Sheffield. If you're listening on YouTube, we'll put all their socials and their wiki in the description. These guys are absolutely charming. Go and check them out.
Oh,
how sweet.
You
I guess
guys,
we'll have to send you all that now.
You guys are charming.
Oh,
No.
so wholesome. Yeah, the wholesomeness comes at the end of the podcast.
Oh yeah,
Absolutely.
We'll
Always listen to
don't
you.
worry. We'll end the recording now and just be horrible to each other.
Yeah, that sounds good.
That's all we have time for today. As always, thanks to our wonderful guests, and please feel free to follow us on Instagram at exeter.igem2022, on YouTube at exeter.igem2022, or pop us an email on exeter.igem2022@gmail.com. Please ask any questions via the social media or in the comments, and any other feedback is greatly appreciated. Bye for now.